Hello, everyone. My name is Pavan Kumar, and I'm a rising senior at American Heritage. This summer, I had the opportunity to work in the Fitzpatrick lab, where I worked on the creation of a software pipeline for cell tracking across learning stages. Well, first of all, what does the Fitzpatrick lab study? The Fitzpatrick lab focuses on studying the structures and functions of the visual cortex. First of all, the visual cortex is responsible for the processing of visual information. And as Ariel said, one of the animals we use in this lab is a tree shrew. We use the tree shrew for two primary reasons. One being that it's a very close relative to the primates, and another being that a tree shrew has a strong visual system compared to other lab animals such as mice. A special feature of the tree shrew's visual cortex is that cells which produce a response to a specific visual stimuli are all clustered with each other. For example, if you look at a visual representation of the tree shrew's primary visual cortex, which is on the right here, we see that tree shrew's cells are clustered in groups which are responsible for producing the same function. So for example, let's say a tree shrew sees a horizontal line visual stimulus. All the red clusters of cells in the brain would be responsible for producing a response. However, let's say a tree shrew sees a vertical line visual stimulus. All the cyan parts of the brain would be responsible for producing a response to that specific visual stimulus. But how do we actually measure these responses? To understand these responses, we take recordings using fluorescent sensors. And whenever there's activity in the visual cortex, the cells that produce the activity light up with the fluorescent sensors. For example, in this video, tree shrew is presented with a vertical line visual stimulus. And when that vertical line pops up for the tree shrew, the clusters of cells in the top right will light up. So as you saw here, the tree shrew sees a vertical line visual stimulus, and then the group of cells in this right area light up. This tells us that there's activity, and this data can later be analyzed to understand learning across different stages. To understand these changes in learning, we can measure learning-related changes in the visual cortex when we perform a visual discrimination task. In our case, a visual discrimination task is when you train a tree shrew to pick out a specific visual orientation in our experiments. If we look at the example on our screen, we want the tree shrew to pick out the vertical line visual orientation, and anytime it picks this orientation, it gets a juice reward. However, if it picks a wrong orientation or a distractor orientation, it gets no reward. This essentially creates an association between a specific visual stimulus and a reward, and over many training sessions, a tree shrew will become an expert at this specific task. Performing these experiments while we image the activity in the visual cortex has shown that there are indeed changes in neuron activity due to learning. The image on the left is an imaging session pre-learning of a tree shrew, while the image on the right is a post-learning imaging session of a tree shrew. Both these imaging sessions have the same two cells, cell 1 and cell 2. However, if we zoom in on the activity of cell 1, we can see that its activity pre-learning is actually significantly lower than its activity post-learning. This tells us that, and this allows us to think that there are actually certain cells that change their activity after the process of learning. And while we know that certain cells do change their activity during the process of learning, we want to know what occurs between these two states of pre-learning and post-learning, and we want to understand why and how these things occur. So currently, to understand this, we perform visual discrimination training tasks across multiple days and actually record the activity in the visual cortex of tree shrews. These sessions normally average from about 30 to 60 sessions of training, and each session has about 300 to 400 cells on average. It's important to track the same exact cells across all the sessions to understand how the cells are changing during the entire process of learning. If we do not track the same cells, we won't be able to make informed predictions as to how the process of learning is actually taking place. This is because two cells likely have different initial activities, and it wouldn't be right to compare two cells that are not the same. And as you can probably tell by now, comparing hundreds of cells across many sessions can become tedious extremely quickly. And this is where my summer work comes into play, where we created an automated software pipeline to help solve this issue. So first of all, a summary of our pipeline. The first step of our pipeline is actually loading in the imaging session data. This step is pretty easy, as we already have these recordings as scientists perform their visual discrimination experiments. Then we just load this data into our pipeline. After the imaging sessions are loaded, we can then perform cell detection, or in other words, finding what cells are present in a specific session. After this step, we can then perform cell tracking, or in other words, finding what cells are present in all future sessions, and then scientists can do their analysis from there. So moving on to our first software module, we use the open source software called Sweet2P for our cell detection. This was developed by Marius Pachitario. Sweet2P uses both an anatomical as well as an activity-based segmentation. An anatomical-based segmentation refers to the picking out of cells based on visual characteristics such as brightness, pixel ratio, and diameter. However, Sweet2P also incorporates an activity-based segmentation. This is the picking out of cells based on activity from the fluorescent sensors recordings. As we saw before, when the fluorescent sensors light up, this tells us that there's actually activity in the cell. And in theory, only a cell should be actually producing those kinds of activity. So the fact that Sweet2P combines both an anatomical with an activity-based segmentation gives us a robust way of finding cells within a specific session. This Sweet2P software is run for all sessions independently, and then we can eventually perform cell tracking or finding which cells are present in all sessions we want to compare. So our next step is cell tracking. And for this step, we use open source software called CellReg. This was developed by Lauren Shentuck. The first step of our pipeline for CellReg is actually loading in the cells. However, this step is already automated as we already get them from Sweet2P. Then we can perform image alignment. In this case, image alignment is when we have all the imaging sessions we want to compare in the same field of view. These alignments are important to compare in the same field of view, as it's really difficult for both the computer and a human to actually pick out cells if there's large shifts in the field of view. The reason these shifts in the field of view even occur in the first place is because tree shrews can move slightly during experimental sessions, and they're not placed in the exact same sessions from day to day of imaging. After we perform this image alignment, we can then perform probabilistic modeling, which uses both spatial correlations as well as centroid distances to come up with a probability that a cell is the same across multiple sessions. We call this value p-same, or the probability that a cell is the same across all the sessions we want to compare. And lastly, I applied a clustering procedure based on the estimated probability of the previous step, the probabilistic modeling step. So first, we created an initial list of cells based on a probability threshold. From our playing around with the parameters, 0.5 worked well. The iterative clustering procedure was then applied, and each cell was clustered with the candidate from which it had the highest probability of being the same as in the subsequent sessions we want to compare. And this was run until all cells were found or not found in multiple sessions. And after that, we produced our output, which can be used both for statistical as well as data analysis purposes. So looking at some of our figures, our pipeline produces about 15 figures, which can be used both for statistical and data analysis purposes. So this is one of our many outputs. And as you can see on the non-aligned image here, the red cells are from session 1 and the green cells are from session 2. And if you look at this image, there's not many overlapping cells. And if you look into a certain area in this image, let's say over here, it's really hard to tell which cells are actually the same, and there's really no overlap. So we're going to have a really hard time picking out which cells are the same. However, we do perform alignment, and alignment image is on the right. This has the same cells from session 1 and session 2. However, after the alignment, you can actually see that a lot of cells are overlapping, and this tells us that they likely are the same across multiple sessions. And after we perform our probabilistic modeling step and clustering algorithm, some of the pairs of cells that aren't already matched up after the alignment will likely match up and will be considered the same across multiple sessions. Also, this data can be used for quality assurance purposes in the sense that if a lot of cells are being cut off, let's say in this alignment phase in this right area, it likely means that there are too large of shifts in the field of view, and it likely isn't wise in the first place to even compare these 2 days of imaging. And this is the final output from our software pipeline. This image on the screen shows the cells that were present in the first day of imaging. All these cells were present in day 1 of imaging we want to compare. However, only the green cells were present in all subsequent sessions of imaging that we want to compare. And this image can be used for further analysis, and the green cells can later be analyzed and to understand how the cells are actually changing across the process of learning. So focusing on some of the advantages of our pipeline is fully automated in the sense that it requires no manual input other than a person loading in the data and pressing run. It's about 25 times faster than manual tracking. For a person to pick out cells and actually track them across 3 to 4 sessions on average, this takes about a full day's worth of work, or about 8 hours, while our pipeline can do the same in about 20 to 25 minutes. We also have more accurate cell detection. A human can normally only rely on anatomical features on the mean image to pick out cells. However, our pipeline incorporates that aspect as well as an activity aspect to actually pick out the cells, so we will have more accurate cell detection. Our pipeline is also reproducible. If 2 scientists have the same data set and they run our pipeline, they will end up getting the same results as is done by a computer. However, if a person was doing this, it's extremely unlikely for 2 scientists to get the same results if they're comparing hundreds of cells across 30 to 60 sessions on average. And lastly, we also get some quantitative metrics from our pipeline, which tell us how well this pipeline is working for the data that's inputted. If it's not working well, it tells the scientists that their data is probably not a good fit for a model, or some parameters need to be changed. And looking at the future and some future things we could add to our pipeline, this includes adding more functionality to 2-channel data, as well as making our program more robust for larger session counts, for example, making it more efficient for session counts comparison that are larger than 40 or 50 sessions. Thank you. Thank you to Gabriella for helping me through every step of this project, Alaria for giving me this opportunity, Erica, everyone in the Fitzpatrick Lab, my fellow interns, and the MPFI supporters who made this internship possible. Thank you.
So as Ariel, I want to say it was 6 centimeters. In the case of Pavan, we go from 8 hours of work of one scientist to 25 minutes of computer working.
Yep.
So I think it's really incredible. Questions for Pavan?
Did I hear you correctly that he did this project and then you did something with the computer that you're going to have at the open house?
Yeah.
Yep.
You did both?
Yeah.
Oh, that's, yeah, great.
Thank you.
Yeah,
What
apparently it
high
was not enough to do this.
What high school do you attend?
The question was, what high school do I attend? I go to American Heritage down in Boca.
Oh, okay.
Yep.
Thanks.
The alignment process is manual, then the automated starts, or is the alignment automated as well when you had to align?
Yeah, so the question was, is the alignment stage also automated? And yes, it is automated. All we have to do is just load in the data, which is done manually. But after that, all the steps of the pipeline is done automatically.
I know. You said that when you're picking out the cells, you use both morphological data and the activity data. So does this mean your algorithm only picks out cells that are actually active, or does it also pick out ones that are not active and just kind of earmarks them saying this cell just didn't do much?
Yeah, so the question was, do we only pick out active cells? So some cells are not as active on all days of imaging. So the anatomical aspect and the morphological aspect picks up those cells usually, while the activity-based aspect of our pipeline actually picks up those cells that actually produce activity. So we do get both of those cells, and further analysis can actually be done to actually split those apart and actually find out which cells were active and which cells were not. But right now, we do not have that functionality.